Welcome back, everyone. We are now ready to begin our Session 3 Q&A panel discussion. Please continue to submit your question as we move into the discussion. I would like to introduce our panelists. We have Dr. Bin Chen, Senior Staff Fellow from the Office of Generic Drugs. We have Dr. Sami Nazzal, Senior Pharmacologist from the Office of Generic Drugs. We have Dr. Jinhui Zhang, Senior Pharmaceutical Scientist from the Office of Pharmaceutical Quality. We have Dr. Maotang Zhou, Division Director from the Office of Pharmaceutical Quality. We have Dr. Keduo Qian, Chemist from the Office of Pharmaceutical Quality. We have Dr. Dahui Liu, Senior Pharmaceutical Quality Assessor from the Office of Pharmaceutical Quality. Thank you to our panelists, and let's take our first question.
Thank you. Thank you so much to all of our panelists. The first question is for Dr. Sami Nazzal. This question is with regards to demonstrating API semaglutide sameness. What are the requirements for the oral formulation, perhaps relative to the injectable ones? More specifically, apart from the primary sequence, is there an expectation to demonstrate sameness in secondary, tertiary, and quaternary structure and the aggregation profile? Many of these parameters become relevant, measurable when API is dissolved. Is there an expectation to provide side-by-side comparison of these attributes upon tablet dissolution in the QC media or other media?
All right. Thank you for the question. That's a fantastic question. There is a language in the PSG that talks about recommendations for demonstrating API sameness and comparative impurity assessment at product level. So the expectations is that applicants to provide information on primary sequence to support API sameness. At this time, there are no recommendations for an applicant to demonstrate secondary or higher structures or any aggregates during dissolution. So it is expected that for dissolution, we are collecting concentrations of the API, and we are not expecting any comparative analysis during dissolution studies. And yeah, I hope that answers the question. Thank you.
Thank you so much, Dr. Nazzal. Our next question is for Dr. Dahui Liu. For colloidal iron products, you emphasize that DLS particle size comparative testing should be performed over serial dissolutions, not just a single concentration. The PSG also requires a PBE statistical comparison for the in vitro BE study using particle size distribution. Does the PBE study need to be performed at multiple dissolutions or just at a single optimal dilution?
So, yeah, so for PBE study, you only need to, for the optimal dilution, it's a single dilution. For the PBE also, but for the method development, you'll do some dilution to figure out the optimal one. What I discuss is for the physical chemical characterization. Thanks.
Thank you so much, Dr. Liu. Our next question is for Dr. Bin Chen. This question is related to the regulatory decision-making process. If the proposed generic drug is subject to bio-waiver for a BE study as described in the CFR, does the applicant need to request for a BE waiver prior to submission of its ANDA? Or can the applicant submit its original ANDA and the BE study will be automatically waived because the sponsor believes the CFR criteria are met, thus no separate request for a waiver is needed?
Thank you for the question. I think this question is actually related to like parenteral solution or ophthalmic solution. The BE is self-evident if the formulation, the inactive ingredients are Q1 and Q2 the same. So I think here for applicants' consideration is that if you are very sure, for example, your formulation compositions is Q1 and Q2 the same, you can just, in your ANDA application, you can just demonstrate Q1, Q2 sameness by your formulation composition table and request for bio waiver because it's self-evidence. You don't need to pre-submit a waiver request prior to the ANDA submission. But if you are not very sure about whether your formulation composition is Q1 or Q1 and Q2 the same, we encourage you to use control correspondences to request the agency to evaluate your proposed formulations, and you can include those communications in your ANDA submission. Thank you.
Thank you so much, Dr. Qian. Our next question is for Dr. Maotang Zhou. When the discussion is about API sameness, does the agency actually mean identical and not similar? Since it's impossible to capture RLD true variability based on limited number of batches analyzed, what statistical can be used to demonstrate similarity?
Thank you for the question. So when we're talking about the API sameness for complex mixtures, we are not talking about identical, but it's not also similar, but the word we use is equivalent. So basically, even IOD itself, you have, in my presentation, I mentioned that even for IOD itself, there are still batch-to-batch variation. So when you develop the generic drug, you can study the IOD batches to capture those variation and statistically meaningful variation. Then you can demonstrate your own, develop a criteria to demonstrate API sameness.
Thank you. Thank you so much, Dr. Zhou. Our next question is for Dr. Keduo Qian. For PPS, the PSG does not specify that the manufacturing process should also be the same for the sameness. Is there a gap between the PSG and the FDA requirements?
Thanks for the question. So the PPS does not require the manufacturing process to be the same as the RLD, and there is no way to know the RLD's manufacturing process. So what we mentioned is the understanding of the manufacturing process is very important, and the process may have left process signature or structure signatures. What we request is the API to be the same with the RLD. Thank you.
Thank you, Dr. Qian. Our next question is for Dr. Jinhui Zhang. What are the critical considerations selecting internal standard for semaglutide?
Thank you for the great question. I can talk for the whole hour, but long story short, let's start with ICH-M10, Bioanalytical method validation and sample study analysis guideline. So the guideline recommend using stable isotope labeled internal standard. But for semaglutide, which has a molecular weight of around 4,000, there's some special consideration. As I just mentioned, if using D8 internal standard, there might be specificity issues that must be taken into consideration. And also for peptides, deuterated labeled internal standard might have different retaining time, which might not efficiently tracking matrix effect. Thus, also need to be taken into consideration. There are 3 commercial valuable internal standards for semaglutide. There's D8, D15, and C13 and N15 labeled, and our in-house research are doing some comparison. So that's pretty much the matrix effect and specificity and the return time. That's the 3 key important need to be taken into consideration. Thank you.
Thank you, Dr. Zhang. Our next question is for Dr. Bin Kin. Is it possible to invite the generic API of a finished product manufacturer representative to this meeting as well to discuss their challenges in getting complex products approved?
Thank you for the question. We always welcome industry's feedback and perspective on this type of topic. We would like to invite a generic representative from either API or drug products to participate in our workshop organized by FDA and CRCG, the Center for Complex Generics. As this type of workshop can help us to understand the practical challenges and getting the feedback on our thinking. So if anyone is interested to attend, please go to the CRCG website. And then we also have a few workshop in plannings for next year. The information of the POC are on the website. You can directly reach out to the POC if you are interested in participating in these workshops and give presentation and provide your perspective. Yeah, thank you.
Thank you, Dr. Qian. Our next question is for Dr. Dahui Liu. At what stage in generic development should sponsors engage with FDA regarding their characterization strategy, particularly for novel iron products that may not have established precedents?
Yes, so there's several, the sponsor can interact with the agency at several stages. Before the end application, the sponsor can interact with agencies through product development meetings such as that to present the strategy and get input from the agency. And after they submit the ANDA, then there's a post-CR clarification meetings and also post-CR scientific meeting. And if it's qualified, they can have a mid-cycle enhanced meeting as well. So those are the scientific meeting, the enhanced meeting, they're more scientific discussion. So the sponsor can use those. And also before submission, the sponsor can interact with the agencies to control the correspondence as well. Thanks.
Thank you, Dr. Liu. Our next question is for Dr. Jinhui Zhang. What are the recommendations for impurities analytical procedure validation in ICH Q2R2 guideline?
Thank you for the great question. ICH Q2R2 was finalized November 2023. There's a lot of very useful resource in Annex 1 and 2 of ICH Q2R2. And Annex 1 basically outlined the validation parameters need to be finished based on the purpose of the analytical procedure. And in Annex 2, there's 9 tables analyzed a lot of very detailed recommendation for different technologies related to impurities in semaglutide. The table on relative quantitation by LC and the quantitation of trace impurities by LC mass is the most relevant. There's a lot of detailed recommendation based on the principle of method. So developer can, that's really something developer can take advantage of. Thank you.
Thank you, Dr. Zhang, for that response. Our next question is for either Dr. Keduo Qian or Maotang Zhou. You mentioned the use of several lots of RLD, more than 3, for establishing API sameness. Understanding there is variability across the RLD, does the OGD have an appropriate number of RLD batches in mind?
So the PSG has a recommendation of the minimal RLD batches should be tested, which is 3. And we understand there are variability in the RLD batches. So it's to the applicant's advantage to test more So we do not have a recommendation of appropriate numbers, but the minimums are 3. Thank you.
Thank you, Dr. Qian. Our next question is for Dr. Sami Nazzal. Today, you covered the PSG for oral semaglutide. I have a question regarding semaglutide for injectable products. Is it also acceptable for recombinant generics for semaglutide for injectable to be submitted under the generic pathway?
This is a fantastic question. So this question is about injectable products. Now, before I answer this question, I need to highlight something that we have a guidance document, and it's actually in front of me. I'm reading the title. It's on peptides and as for certain highly purified synthetic peptide drug product. Now, in that guidance, there is a footnote that says that for injectable products, if an applicant wants to pursue a peptide of an rDNA origin, that perhaps they may need to pursue a 505B pathway because they may need to perform clinical studies to assess the host cell impurities. However, with that being said, and this is an important point to applicants interested in pursuing a generic product of a peptide of rDNA origin, is that if an applicant can justify that the risk of host cell impurities are controlled, and if perhaps they can demonstrate through maybe in vitro studies that the risk of impurities or that is no greater than the RLD, then it might be permissible to pursue an ANDA 505J pathway. However, we advise and recommend for potential applicants to reach out to the FDA through a pre-ANDA meeting to discuss their strategies before they submit their application. Thank you.
Thank you, Dr. Nazzal. Our next question is for Dr. Maotang Zhou. You listed improper negative control studies as a common deficiency. What constitutes an appropriate negative control for GA characterization methods, and how should we design these studies to properly challenge the sensitivity and specificity of the analytical methods?
Thank you for the question. So the purpose of negative control is to determine, to challenge your analytical method, specificity or sensitivity. So we see some examples that these samples of test product are obviously different from the generic. For example, the amino acid ratio are different, components are different, or molecular, even the apparent molecular weight are different. So those are not enough, sensitive enough to test the challenges your analytical method. So ideally, you should have something with a similar component, amino acid components, and also similar, molecular weight, but you have some minor key difference in structural signature. That way you can challenge your method. Thank you.
Thank you, Dr. Zhou, for that response. Our next question is for Dr. Bin Kin. How to establish Q1 sameness if a drug product is made of more than one PLGA polymer? Do we need to separate the polymers and characterize them individually?
Thank you for the question. For PLGA based product, if the product contains more than one PLGA polymers, to demonstrate Q1 sameness, the applicant only need to extract the polymer and categorize the polymer as a bulk, which means that they don't need to separate the individual polymers and categorize them individually. Thank you.
Thank you, Dr. Kin, for that response. Our next question is a follow-up to a previous one regarding the use of several lots of RLD, potentially more than 3, for establishing API sameness. We understand the general recommendation on testing 3 or more batches to understand the RLD's variability. However, when there is an availability issue, is it acceptable to submit data on less than 3 lots with evidence supporting the unavailability?
Thanks for the follow-up question. So yes, when there is availability issue and you submit support, we would still recommend that you provide 3 test batches, even though you have less than 3 lots of RLDs.
Yeah, I want to add a little bit on that. That's right, Keduo is right. So the availability is a real issue, and then you can submit less than 3 batches with your justification. Like Keduo said, to test more batches LD is for the sponsors' benefit. The number of test product, it has to be 3 batches. The numbers of RD batches actually depend on the purpose again, right? So sometimes for the physical chemical characterization, if you do see a difference between the LD and the test, you may want to test more batches of LD to justify those differences. Thanks.
Thank you, Dr. Qian and Dr. Liu. Our next question is for Dr. Jinhui Zhang. In your slide number 7, there are data using 3 m divided by z value presented. What is the advantage of using 3 instead of 1?
Okay, that's an excellent question. So we were using high resolution mass spec for data for impurity profiling and quantitation. So in mass spec data, in high resolution mass spec, all the peaks are mono isotopic peak. So different isotopic distribution can be readily resolved by mass spec. So for different peak have different C13 component. So we use 3 different MOZ can cross-reference each other to make sure the specificity. That's also part of the recommendation in ICH Q2R2 and XVII. Thank you.
Thank you, Dr. Zhang, for your response. Our next question is for Dr. Sami Nazzal. Published research showed semaglutide absorption occurs through a highly localized transcellular mechanism in the stomach. How does this unique mechanism influence FDA's bioequivalence approach compared to typical oral drugs?
For majority of, for orally products given by the oral route, we do recognize that most of the absorption happens through the GI tract, probably small intestine. Now, in case of semaglutide, it's very unique because it has this localized absorption in the stomach. And we recognize that. And Jinhui, he gave a fantastic talk where he talked about the issues related with SNAC and its mechanism and how it works and different variables. And we recognize that a lot of this is already published. So we took that into consideration. So, for example, in option. Well, first of all, I'm using language from the PSG. We require that to establish similarity, there is a solution requirement where we are asking applicants to perform solution on both to measure semaglutide as well as SNAC. And because we recognize the importance of SNAC as an important ingredient in the formulation is when we do this, when I was discussing quantitative similarity, that quantifying the amount of SNAC in the formulation is very critical. So that is basically how we take that into consideration. I hope that answered the question. Thank you.
I want to add a little bit to Sami's response. It's a great response. I want to talk a little bit about the absorption kinetics and the site of absorption. So as we just discussed, the three major mechanisms of SNAC enhance absorption is to enhance permeation and prevent semiconductor forming oligomers, and also the buffering effect to increase gastric pH. So if we believe it's passive diffusion, so the concentration gradient is very, very important, right? So that's the reason we mentioned a couple times, synchronized and fast release of both semaglutide and SNAC is the key. And also you can find from the label of oral semaglutide tablet, there's one recommendation that using 2 milligram strength tablet to replace 140 milligram tablet is not recommended. That means a lot. Then I'm going to talk a little bit about site of absorption. As Sami said, many drugs absorbed in small intestine, but semaglutide is different in stomach. So all the recommendation in the PSD option 2, option 1 requires follow the administration instruction of RRD, which is fasting and using less than 4 ounces of water, right? So all those kind of recommendations based on the fact that absorption in stomach. If the generic develop using different enhancer to design the product absorbed in intestine, so that will be a issue. So we strongly recommend the developer to discuss with the agency upfront. Thank you.
Thank you, Dr. Nazzal and Dr. Zhang. Our next question is for Dr. Maotang Zhou. The presentation highlighted that ultrafiltration, diafiltration can impact structural signature characterization based on collective analysis. Can you elaborate on specific examples where inappropriate UF-DF conditions have led to ANDA deficiencies, and what fractionation studies you recommend to adequately assess this impact?
Thank you for the question. So, like I said in the presentation, the ultrafiltration step does not involve any formation or break of chemical bonds. So we believe that the structural signature or process signature established during the previous step, such as the polymerization or a depolymerization step. But since the characterization measure collective or aggregated factors of the polymer, so that it's going to impact the determination of the API sameness. So one example of the inappropriate use of the ultrafiltration characterization is, for example, if your molecular weight, average molecular weight is not acceptable, but you use ultrafiltration as a control to make it meet the requirement. That way is not acceptable. So we recommend that you use a fractionation study to study the structure signature before and after the ultrafiltration to demonstrate they are identical or equivalent, so that the ultrafiltration step did not impact the structure signature individually. Thank you.
Thank you, Dr. Zhu, for that response. Our next question is for Dr. Bin Kin. For cross-linked polymers such as PMDS and hydrogels presented, Q2 can be challenging. Is it possible to still file or follow the PSG when the Q2 is not within the conventional 5% range?
Thank you for the question. I think for cross-linked polymers, the agency does recognize that sometimes it's very challenging to meet the Q2 criteria using the conventional like plus minus 5% range. So in this case, for the filing purpose, it may be still feasible if the applicant can provide adequate justifications. For example, whether this change can result in any difference in the mechanical properties, whether this can impact the product performance. I mean, in brief, like the agency will evaluate the Q1, Q2 in a totality of evidence approach. We will take all the informations you provide, and the conventional plus minus 5% may not be applied in this case. Thank you.
Thank you, Dr. Kim. Our next question is for Dr. Dahui Liu. Can we use non-USP grade excipients in the formulation while RLD uses USP grade? This is in order to meet the Q3 characteristics of the PSG requirements.
Thank you for the question. Theoretically, if it meets the quality standards, theoretically it's acceptable. But to get more feedback from the agency, it depends on the cases, case by case. The sponsor, you can submit a controlled correspondence with background information to obtain feedback from the agency. Thanks.
Thanks, Dr. Liu, for that response. Our next question is for Dr. Keduo Qian. You mentioned that the structural signatures are molecular fingerprints left by the manufacturing process. For semi-synthetic products like PPS, how can we differentiate between signatures from the extraction/purification of the starting material versus those from the chemical modification steps, and which are the most critical for demonstrating sameness?
Thanks for the question. It's a very nice question. So both are equally important, no matter it's from the starting material extraction or from the chemical modification following the extraction. So as we mentioned, the RLD and the generic PPIs should be equivalent. So this equivalency are the most important. So understanding the process are very critical to develop the products that are equivalent to the RLD. Thank you.
Thank you, Dr. Qian. Our next question is for Dr. Jinhui Zhang. What are common approaches investigating API excipient interaction?
That's a great question. There's numerous different ways to explore the potential interaction between API and excipients. For example, size quotient chromatography is a common way to see whether there are the binding kinetics of API and excipients. And recently, we are working on a relatively new approach called the hydrogen deuterated exchange mass spec to profiling any potential interaction site at the surface of semaglutide. So stay tuned. Thank you.
Thank you, Dr. Zhang. Sorry. Our next question is for Dr. Sami Nazzal. We've seen industry interest in alternative absorption enhancers beyond SNAC. What should generic developers know about pursuing these alternatives?
The topic of alternate absorption enhancers other than SNAC is a very, very interesting topic. It's a very complex and requires very special consideration. We do know that there is some publications out there that talk about absorption enhancers and how they work, and there is a belief that perhaps those absorption enhancers are compound specific. And already Jinhui talked a lot about, for example, SNAC, about the different ways by which SNAC works. So the question becomes, if somebody wants to use a different absorption enhancer, so the question becomes, will that enhancer have similar activity as SNAC, or are they comparable? And also in terms of quantity, is it similar? So there are a lot of questions about those alternate absorption enhancers that needs to be addressed. Now, there's also another question if, for example, if somebody is proposing to use a new absorption enhancer, then perhaps maybe in that case, they may need to pursue a 505B pathway. So there are a lot of questions here, but we do encourage applicants who are interested in pursuing alternate absorption enhancers to reach out to the FDA through either control correspondence or perhaps through a pre-end meeting to discuss their issues and their strategies.
So I want to dovetail what Sami mentioned as actually it's a very, very hot topic. I mean, I just want to revise what Sami said a little bit. It's not some publications, lots of publication on this topic. So for example, some big pharmaceuticals, they're developing GLP-1 GIP DAO agonists. So their design is intestinal absorption. So oftentimes when they publish paper, they use semaglutide as positive control. However, we haven't seen this kind of product approved. So it's amazing, but very, very tough product to develop. And also Sami brought out the point that the mechanism of absorption is really important to determine the regulatory pathway for the prospective product. Thank you.
Thank you, Drs. Nazzal and Zhang. Our next question is for Dr. Bin Chen. Can I submit a Q1, Q2 control correspondence for a drug product where the regulation does not require it to be Q1, Q2 the same?
Thank you for the question. So usually, if for applicant submit control correspondences for agencies Q1, Q2 evaluation, the product needs to be, for example, required by regulation to be Q1 and Q2 the same, or the agency's product specific guidance recommend that. I mean, the PSG recommend the Q1 and Q2 sameness as part of the B approach. You need to meet these two criteria, like to submit control correspondences for Q1, Q2 evaluations. But if your product is not required by regulation to be Q1 and Q2 the same, there is no PSG or the PSG does not have these requirements, and you are proposing an alternative B approach, and you believe Q1, Q2 is an important component for your approach to demonstrate BE, you can submit a pre-ANDA development meeting to the agency for discussion. Thank you.
Thank you, Dr. Chen, for that response. Our next question is for Dr. Dahui Liu. The PSGs for iron products recommend a comprehensive list of comparative characterization studies for supporting BE and product development. What are the general expectations for concluding comparability? If certain characteristics appear to have differences, such as molecular weight or molecular weight distribution, between the T and R products, can the differences be justified? And if so, what are general expectations?
Oh, thank you for the question. This is the main thing for the evaluation of comparability for the characterization. As I mentioned, is a challenging task here. That's why our recommendation is always the totality of the evidence. So like what I said, when there's uncertainty, that's when we recommend you to do orthogonal methods for a certain property characterization to reduce the uncertainty. Normally, actually, when we see during the assessment of the applications, we normally do not see a single difference in one test. Those are the differences, actually, they are all kind of correlated. Sometimes if the method, actually, if a method is very sensitive, it shows the differences. And start from that, actually, when we look back, we can put the puzzles together, and actually you can see the evidence can be corroborated by other test evidence. Sometimes those even can go back to, because those are intertwined, you can even go back to manufacture process concerns there. So again, it is if differences can be justified, theoretically yes, right? So if it's adequately justified, it is. In terms of expectation, we do not have a defined expectation here due to the natures of some of those tests. Again, it is a justification. We see the totality of the evidence. So it is sometimes it is for the sponsor to really how to convince us using your approach. But there's always a door open for you to come here and to provide sufficient evidence. And that's it. Hopefully that can address it. Thank you.
Thank you, Dr. Liu. We have a follow-up question for Dr. Sami Nazzal. This is regarding alternate absorption enhancers. Can applicants pursue option 1 BE studies as indicated in the PSG?
I mean, thank you for the follow-up question. And before I answer, when we were discussing the previous question on alternate absorption enhancers, perhaps me and Jinhui were discussing more about the science of absorption enhancers. It's a beautiful, beautiful topic and a lot of things to discuss here, but I probably missed that point. And thank you for the follow-up, is that yes, there is a possibility that if somebody is pursuing an alternate absorption enhancer, that they can pursue option 1. Because option 2 assumes quantitative and qualitative sameness and similarity. So it will use absorption enhancers that's different from SNAC, then probably option 1 might be considered. Thank you.
Yeah, I just want to add a little bit. I mean, thanks, Sami, for clarifying the major difference between option 1 and option 2, which is very, very crucial. And also, I mean, as we just mentioned, a lot of pharmaceutical industries, they're developing different way to make it orally available, right? For example, we saw the papers doing the barcode delivery. If it happens, really amazing, right? And it's bypassed the first pass effect. However, I mean, if you exactly follow option 1, right? For stomach absorption, swallow the tablet, no chewing, with 4 ounces of water, but for barcode, that's totally different story. So again, everything based on the understanding of the absorption enhancing mechanism and the earlier communication with the agency. Thank you.
Thank you to Drs. Nazzal and Dr. Zhang for that response. Our next question is for Dr. Keduo Qian. With the extensive list of characterization methods required, such as NMR, ICP-MS, GCP, and such, do you have any recommendations on how to prioritize these techniques during development? And are there any emergent analytical methods that FDA is considering for future PPS guidance updates?
Thanks for the question. So there is a reason these products are considered complex API or complex products. It's because they are more mixtures or polymers. So that's why a lot of different technologies are needed to address the sameness requirement. So the methods themselves are complementary to each other's. So they're both very important. If you can use one method to address multiple sameness requirement, that would be great. But and also we do not really have a requirement of the method itself. The applicants are free to develop your methods that are suitable for the purposes. Thank you.
Thank you, Dr. Qian, for that response. Our next question is for Dr. Bin Chen. In your presentation, you mentioned PDMS chemistry information should be provided to support Q1 sameness for PDMS, but the PDMS chemistry of innovator product is unknown to the generic firm. How can I test product to use the same PDMS chemistry?
Thank you for this question. For PDMS, there are different curing methods. So for a generic company, the proposed test product may not necessarily be the same to the PDMS chemistry as RD. But we do also need the firm to provide additional competitive categorization, such as cross-linking density and the comparative mechanical property, as well as other information like the dimensional information. We will evaluate Q1, Q2 as taking all the information into consideration. Thank you.
Thank you, Dr. Chen, for that response. Our next question is for Dr. Dahui Liu. Should sponsors include stress stability testing as part of their comparative characterization studies to demonstrate that the test product maintains the same interaction profile as the RLD throughout its shelf life?
Thanks for the question. So this specifically for the iron colloid product. So far in the PSG, we do not add a recommendation to test the product at different age. So at this point, we don't really have specific requirement for the test batch age. However, this will be, other than the physical chemical comparative characterization, for your quality control, you can certainly do stress study to figure out your specification. That will be helpful. And that's it. Thank you.
Thank you, Dr. Liu. Our next question is for Dr. Keduo Qian. Since PPS is derived from naturally occurring botanical raw materials, what are your recommendations on addressing potential variability in the silane starting material across different harvests or geographic sources while maintaining API sameness with the RLD?
Thanks, that's a very good question. So as a product derived from naturally occurring starting materials, they're generically inherited that there will be variations from batch to batch. And the RLD will also have variations from batch to batch. So as I mentioned, the plant species needs to be identical with the RLD. And there are DNA methods that could distinguish not only the species, but the subspecies. And also the geographic areas should be similar or the same to the RLD. And because this product is coming from the plant parts of the wood, the variability will be less. For example, if it's from the leaves, but still those variations should be within the RLD's variations so that equivalence can be achieved. Thank you.
Thank you, Dr. Qian. Our next question is for Dr. Jinhui Zhang. Is it possible to develop other non-parenteral routes of administration for semaglutide?
Oh, great question. So I will start, and then Sami, you can add something. We already discussed about a previous discussion. So a lot of pharmaceutical industry, they are developing non-parenteral absorption through intestine or buccal delivery. However, I mean, we haven't seen any FDA approved this kind of oral product, and they publish their development. And regardless experience gained, all lessons learned will be invaluable to all the generic developer.
There's really nothing I can add. I mean, I'm speaking on behalf of myself as a scientist. I mean, it's a fascinating topic, and yeah, we look forward to see what's going to be published and what comes out. Yeah, Jinhui.
Thank you, Drs. Nazzal and Dr. Zhang. Our final question for today's Q&A panel will be for Dr. Sami Nazzal. Why studies on R1 cannot be waived based on R2 studies?
Now, to answer that question, I encourage applicants to look at we have a guidance we call the General BE guidance that provides a lot of information about BE studies, and there are some language in that guidance about waiver for products of different strengths. So part of the presumption here is that there should be some degree of formulation similarity, meaning using similar excipients. But in this case, in semaglutide R1 and R2, they are different formulations, which is why they cannot be waived. So R1, to answer the question, cannot be waived based on R2 because of these differences. Thank you.
Thank you, everyone. That concludes our Q&A panel discussion for session 3. Thank you to our speakers and panelists for an insightful discussion. We will now pause for lunch. Please rejoin us at 1 p.m. Eastern Time as we begin session 4 on nitrosamines, known issues, and practical advice. We look forward to seeing you after the break.